I'm from Washington University in St. Louis, and I will be the moderator of this ADPD 2017 forum that is a phase 3 forum concerning the race for the first disease-modifying drug for Alzheimer's disease. Now, some of you have been to previous forums in this meeting, but just in case, a couple of housekeeping notes. First, please take your phone and put it on silent mode so it doesn't disturb others. And second, if you have not been at a forum, you use your smart device to go to the ADPD app, find this forum, click on that, and then you'll find a notation for Ask the Speaker. And that's where you type in your question that you have for any or all of these speakers, and we will try to get to as many of the questions as is possible. Cannot guarantee that we'll get to all questions. But please be free to ask any question as long as it's related to scientific issues. We think we're aligned, arranged up here in the order you see on the screen behind us. And it's a real honor to be able to participate in this forum with such a distinguished group of panelists. They're going to introduce themselves and give a very brief introduction about what they think the factors are for success in phase 3 trials for Alzheimer's therapeutics, and then we'll open it up for questions. So I'm first going to introduce Larry Altstiel.
Thanks. I'm Larry Altstiel. I'm the Chief Medical Officer for VTV Therapeutics. We are a clinical stage company and are involved in a phase 3 clinical trial in Alzheimer's disease, actually completing enrollment in the second of our 2 pivotals. We target something called the receptor for advanced glycation end products. It's involved in many of the pathologies that are salient to Alzheimer's disease, coming off a phase 2 study with showing that we had some significant progression or effects on disease progression in mild patients after about 18 months. After about 18 months. And so we're embarking on our phase 3. We're finishing up enrollment on our phase 3 program and running under a special protocol assessment and also a fast track status with FDA.
Thank you, Larry. So my name is Samantha Budd-Haeberlain, and I'm the head of clinical development for Alzheimer's disease at Biogen. I've been with Biogen a couple of years, but in total I've been in industry for now more than 17 years, first on the research side, then into translational medicine, and now into the development side of industry. And like many of the people in this room, I also did research in academic centers, postdocs and otherwise, prior to joining industry. But I have to say, sitting here among my august colleagues here in clinical development in Alzheimer's disease, I really love what I do. The title we have, The Race to the First Disease-Modifying Treatment in Alzheimer's disease. When you ask yourself, so why is it a race? It is because there are no disease-modifying treatments in Alzheimer's disease today. And when I come to a conference like this, and I see the advances in science and all the new targets and mechanisms that are possible to be able to become new therapeutic pathways in Alzheimer's disease, it's very exciting. But I have to be a little bit humble and say that from the development side, we're not quite as advanced. We're still creating the ways and means to be able to test those new therapeutic modalities. Now, don't get me wrong, we have actually learnt an incredible amount the last 10 years. Not least, one of the biggest learnings that I believe is very important and shapes the way we do clinical trials today is the understanding that Alzheimer's disease starts long before the appearance of symptoms. And so that poses a very particular challenge to us doing clinical trials in Alzheimer's disease, which is how do we take especially those pathways that we're most familiar with, amyloid and tau, that potentially have a role early in disease, how do we, in the clinical setting, test those new molecules and determine efficacy in a clinical setting. Now, there are some really exciting and bold efforts out there in at-risk individuals who are cognitively normal. So we have API and we have the A4 trials. And those are extremely exciting, but rather large undertakings. But they have also shaped most of the clinical trials that we're conducting today. So if you look at probably the trials of everybody on this panel, we are looking at patients much earlier in their course of disease than was previously done. Quite a big shift in the last 5 years. One of the other important factors about a race implies competition. However, I think the learnings that we have had in the last 5 to 10 years from across the companies here and some of the others present, such as learning much more about target engagement from the Roche, the Pfizer, Janssen studies, learning absolutely the correctness of diagnosis from the Lilly and, again, the Pfizer studies, have been huge success factors. And you see them now almost routinely implemented in our trials. So we have come a long way, but I give a great kudos to my colleagues here sitting on this stage, because what we do is much akin to flying a plane, but also building it at the same time. I think as a final critical success factor, I would say, is actually collaboration. This isn't a competition, and we do need to continue to share our results so that we can learn from each other at conferences like this.
Okay, excuse me, just before we go to Paulo, I know that Samantha wanted to, and brought up very correctly, the move now to at-risk trials, and she named the API and the A4, and of course what she also wanted to name was the Diane Trials Unit, so I just... All right, Paulo.
Hello, my name is Paulo Fontoura. I'm the head of clinical development for Neuroscience at Roche, and I've been with the company for just about 9 years, coming from translational medicine, early development, and now late-stage development. And as you probably know, Roche has been in this race, if you want to call it that, for a number of years now. We currently have 2 molecules in phase 3 development trials, 2 monoclonals targeting amyloid, which are just starting a new generation of phase 3 studies based on the learnings we've all had from the past years from other efforts which have been going on. And unfortunately, all those efforts have brought us a step further, but success still has not really come. As a company, you probably know that very core to our DNA is the emphasis on biomarkers and testing. So we were also very involved in developing CSF diagnostic biomarkers, PET tracer imaging, because we truly feel that it's only by correct diagnosis and correct identification of players, of the patients that will participate in these trials, can we be successful. So that alone has been a huge change in the field, the way I've seen it for the past 9 years. The fact that now it's almost inconceivable that you start a new clinical trial in Alzheimer's without correctly identifying the patients for those trials. Another success factor, as Samantha mentioned, was this move to go earlier, and we're really here in the face of exciting new developments, not only in trials for sporadic Alzheimer's, but also in the efforts that we are doing together with API and Diane to test people at high risk, or actually familial forms of Alzheimer's as well. So reflecting, and I think that should be the topic for today's discussion, about what is it that we still don't have, what are we missing? And my personal reflection on this is that as a research field to develop new medicines, we're still extraordinarily slow and inefficient. It takes too long for each one of us to learn from the others. It took 5 years of the previous generation of trials for us to learn about the importance of early diagnosis, about the importance of biomarkers, about the importance of endpoint selection. And I hope it doesn't take another 5 years to us to learn the next generation of difficult lessons. And therefore, until we really improve the efficiency of that R&D engine, it will be very hard for all of us to make significant moves forward. I don't want to leave us on a sour note, so I do believe in the power that science have to solve complex biological problems, and the fact that now we are starting to identify the predictors that put some patients at much higher risk of progression, I do believe will be a critical success factor for trials in the future.
Be on.
Thanks, Paulo. So I'm David Michelson. I'm responsible for clinical research at MSD, as we call it here, Merck in the United States, on the neuroscience side. And as most of you know, probably, we've been running several large phase 3 trials with verabecestat, which is a BACE inhibitor, one of them in patients who are already symptomatic, one of them in patients with prodromal Alzheimer's. And so, unfortunately, really, the first of those studies stopped just a few months ago, stopped by the DMC, the external DMC. And the second one is ongoing. They recommended that that one continue. So, which brings me a little bit to the title, and I was thinking about, being asked about factors for success, and it feels a little bit like hubris on my part in the context of having just stopped a study to opine on success. But I'll take my chances and go ahead. I guess it seems to me that there are a couple of things that we really need to move forward, and maybe the most important of those, I think, is to continue to explicate and better understand the biology. And what I mean by that is really what drives the hypothesis that we test in the clinic. So if you can have the best designed study with the optimal biomarkers and everything else, and if you don't have a good hypothesis, if you don't have the right hypothesis, ultimately you won't have a positive study. So really continuing to understand and further elucidate the science in a way that informs the hypotheses we test in the clinic, I think is critical. The second piece has to do with tools and methodologies, which is, again, these continue to evolve. We've learned a lot, but they need to continue to evolve. That is, we need to really understand how best to do the studies in order to detect a signal if it's actually there. So it's really a kind of a critical thing. And then the third one I'd say is really around urgency. And I think this is kind of a double-edged sword. So it's obviously, it's a pretty horrible thing to have Alzheimer's. It's a pretty horrible thing that there are as many people in the world who have it as they do. And it's critical for us to feel the urgency to do the work that will change that. At the same time, I think it's critical for us in the context of drug development to make sure that that urgency doesn't overwhelm the ability to do the studies in a way that can get us to the right answer. So finding that balance is really, I think, a critical piece of the work for us. So with that, I'll stop and hand over to Andy.
Thanks, Thanks, David. So I'm Andy Satlin. I'm from Eisai Pharmaceuticals. Eisai is the Aricept company. You all remember Aricept, right? Aricept is from the days when we knew how to do clinical drug development. So everybody knows the problems today are that we're failing in clinical trials. And that's not really a surprise. As everyone has pointed out, Alzheimer's disease is a very difficult disease, a lot of challenges. The brain is a very difficult organ, even when it's healthy. So it's not a surprise that we're failing. The problem is that we're failing too often in phase 3. And that's a big problem because it takes a lot of time and it takes a lot of money to fail in phase 3, and that's wasted time and wasted money. So we need to do a better job of development prior to phase 3 as an important success factor for this field. And we need to learn more about the clinical effects of the drugs that we're taking forward. We need to learn about the appropriate doses to take forward, the endpoints that we need to use, the populations that we should be studying, how long we should be treating, et cetera, et cetera. So it's difficult to do good phase 2 in a complicated disease like Alzheimer's. But there are methods that can help us to do that. And one approach that we've taken at ASI is to do a Bayesian adaptive design in phase 2 that allows us to plan for a relatively large trial, but to take frequent looks at the data on an interim basis and make decisions with regard to changing allocation of patients to different doses, even looking at how endpoints are progressing in terms of variability and other factors as well. And to possibly enable us to get a signal of success or futility early so that we can avoid throwing resources and money after a treatment that might not end up being successful. And it's an approach that we're now taking also with the IMI EPAD project, where the proof of concept study in that project is also using a similar type of design. That'll be a platform trial that'll allow us to put drugs from different companies into the trial on a rotating basis once we get started early next year. There are other aspects of that project that will be helpful to the field as well, we believe, including setting up a very experienced trial network and a longitudinal cohort study that will supply well-characterized patients in the preclinical and prodromal stages of Alzheimer's disease for the proof of concept trial. So I think we need to look at things in an innovative way with regard to actually how we're doing clinical drug development and hopefully use that to avoid some of the failures that we've seen, unfortunately, in this area. Thanks.
Thanks, Andy. Hi, I'm John Sims. I'm the Senior Medical Director at Eli Lilly, and I'm responsible for the AZ Lilly base compound that we're testing now in phase 3 in early AD. And we have in our area, our work area, a couple of quotes from Thomas Edison when I was thinking about this, when we were supposed to think about factors of success. And it's been sitting up there for a while now, and one of his quotes is that I have not failed. I found 10,000 ways that it doesn't work, which is about what we are looking at, aren't we? And I think everyone up here on this panel knows that when we go currently and test a drug, we're actually testing two things. We are really testing trial design, and we are testing the drug. And right now, we are not so clear as to which plays a role in failure. And there are about 10,000 variables in these large phase 3 trials, and there are ones that you know very well, such as the size of the trial, the patient population that you choose, the entry criteria, but there are a lot of other variables like country selection, site selection, all these play key variables. And of course, the primary outcome is still not well known and understood. So every time we do this, we have to learn from the failures. We have to be able to share that data. And that is certainly our goal, is to share some of the most recent failures with solanezumab and get that data out for people to do their own analysis on. And, I think the key also is another Thomas Edison quote, which is many of life's failures are those people who were close to success and then quit. Right, so I think the other thing is we have to keep plugging along, because it is these trials that have failed that have, I think, as Samantha said, we've learned a ton from these. I think we didn't truly know how late is late or how early is early. And I do know that there's a lot of talk about going early, but I usually refer to a lot of other diseases for my education. And I would remind this group that other than infectious disease, it is extremely hard to find an example of a drug that actually showed its first success in a prevention trial. We've heard a lot about going in prevention, going early. Most all the drugs we've ever discovered have made their first success in at least a symptomatic phase. And so I think the question we have at hand is where is this sweet spot? I think we don't truly know this answer. Is it a subjective cognitive impairment? Is it MCI in early AD? I do think that doing prevention trials will be important, but they're not going to be easier. The grass will not be greener, I suspect, on that side of the fence. And I gave a talk a while back ago looking kind of at trial designs and the evolution. I don't know if many people realize it, but there was no such thing as an AD trial before the 1970s. It really wasn't appreciated that senile dementia was AD until the 1970s. And when you look at the trials that occurred in the 70s, the common denominator that I like to use is patient years when we think about trial design. The average patient year of those trials done in the 70s was 4 patient years. When we look at donepezil, its success was 150 patient years. Right, we are currently looking at around trials that are in the 3,000 to 4,000 patient years. Since the AD trial started in the 70s, basically the trial sizes have tripled about every decade. And I suspect if we start talking about prevention trials, we are looking at in the 2020s of possibly trial sizes that could be as high as 15,000 patient years. So this is a substantial issue for us because we, unlike other trials, we're actually studying 2 people, if we're talking about the symptomatic phase, certainly, and even in the prevention trials. So, because we have the caregiver or the study partner, we call them. And so that's really almost 30,000 patient years when it comes to recruiting for these trials. And it is a major burden to learning from failure when it takes so long to recruit these trials. So.
Thank you. Hello, I'm Johannes Streffer, Janssen Research and Development. Thank you very much for inviting me to participate in this very exciting panel. I started my career in Alzheimer in 2000 in a group of Roger Nitsch and Christoph Hock in Zurich, and then moved on from there to the early development in Janssen. Started to care about the base molecule, which we have now taken into late-stage development. So with the molecule, I moved on, and I'm now in late-stage development. I am, and I would like to say we at Janssen, we are very passionate about 2 things in that respect. And the first thing certainly is early treatment intervention, secondary prevention, preclinical AD, and I realized that when Samantha and John mentioned API and A4 and Diane, they wanted to leave it to me to mention the 4th effort in that respect, which is the early trial, which is a very similar design to the A4, but distinctly different. And we do think that we should try to go as early as we can go. As you say, the trial designs are very important. We do think now this trial design is possible, so we've decided to take our base molecule into the early trial, but as well Diane. So these are 2 efforts where we think we can test the molecule and really test the hypothesis if it's working. The other thing we are very passionate, and Annie mentioned that, is the platform trials ongoing. And I would like to mention the platform biomarker trials ongoing, because I do think efforts like ADNI and ABLE have been flagship for the field, telling us how important it can be if there are joint forces. ADNI, I think, is a wonderful experience of joint forces of very strong academic groups with industry, sharing knowledge and sharing design and really sharing to do this. Now, as Andy mentioned, there is now EPAD and there is EMIF in Europe, where we have data initiatives, but as well potentially platform trials in which we can collaborate and potentially have better engines to develop molecules in the future, but that will have to be shown. There's one last thing I would like to remind us all on. This is actually a wonderful conference, and thanks for the organizers getting it together. We have a lot of very interesting science here, a lot of the interaction of amyloid and tau, Lewy body and amyloid plaques. But we have to realize that all the phase 3 programs kicking off today are in principle built on the knowledge of ADPD 2 years back. The ones ongoing are most probably built on the ADPD 4 years back. And when you see the results, and it's been a real good trial, real quick, it's been on the ADP 6 years back, but rather 8 years back. So a phase 3 trial often seems a little outdated the moment it starts, because it cannot, by the nature of it, be at like the interaction of amyloid tau that is currently hot and fancy. But we can try to be as good as we are. And that is, to our understanding at the moment, the population and having a very potent molecule.
Thank you. I'm Terry Fullerton. I am a senior director at Pfizer in global products development. As the only current company that doesn't actually have an asset in phase 3 development, I'm feeling a little bit like a rookie on his first day of training camp, joining a team of all-star veterans here. So I appreciate the opportunity and the organizers for allowing me to participate. So I think I can't offer a lot of anecdote or factors for success from recent experience. Obviously, we believe the learnings from the past, from some of the trials that Pfizer have done, have contributed to today, as Samantha mentioned earlier. I think some of the success factors that I think about as we take, we have a selective BACE1 inhibitor and a gamma secretase modulator that we're poising for, hopefully, late stage development in the near future. The success factors that I think about are really looking inward into our companies to try to put our management at ease as they see these failures arising really on a daily or monthly basis, and trying to communicate the fact that the failures of today really represent the science of 5 or 6 or 7 years ago, and that sort of the pace of scientific advancement has really outpaced our ability to respond in new clinical trial programs and paradigms. I think Andy mentioned the use of greater flexibility in our trial programs and designs through Bayesian adaptive designs. I think that's a great opportunity to more readily incorporate new states of knowledge into our programs more flexibly. We also need to think about how do we best integrate new knowledge into our decision-making process, particularly from the view sort of from the back of the pack, learning from all of the trials that will be coming ahead of us, and how do we best quantitatively integrate that information both into our trial designs as well as into our decision-making framework. So we're thinking very much in terms of quantitative longitudinal disease models and incorporating quantitative treatment effects into our trials and through trial simulation, trying to explore different scenarios to try to best be prepared to respond when the data become available to us. So the last point I'll make really is a point that a few others have made already, is that one of the other success factors that I think that we really need is for all of us to continue to be advocates and champions for transparency of data communication from everything that we learn as quickly as possible. We owe it to the patients, we owe it to the field to get this information out as quickly as we can so that others can learn from our successes and our failures. And I know at Pfizer we've got a commitment for that transparency, and I know that we, as a field, we've been doing a pretty good job of that, and I think that we need to continue to do so as we move forward.
So thanks for each of your introductory comments. I don't know if the audience is aware that our panelists were given really an almost impossible task. They were asked to, without slides, give a 3-minute overview of the factors for success as they see it for Alzheimer trials, and that's a tremendous challenge to do all that in 3 minutes, and more or less they did, so let's give them a round of applause. We have lots of questions already, so we'll jump right in, and I think each of you could anticipate that this question would be asked. Why is the industry persisting in trying to treat A-beta in spite of continuous failure? So who would like to take the first reply for that? Okay, so thank you, Samantha.
So it's a terrific question, isn't it? And why are we persisting? Well, the science is very compelling for A-beta. If we look at the genetics, we look at the animal models, we look at the early clinical studies that we have to date, the information that we've gained over the last few years, it's a hypothesis that continues to be compelling. Now, I think we're closer to getting the right molecules to test if that hypothesis translates into clinical efficacy in patients. And patients is the other reason that we're persistent. This is a terrible disease. It's affecting millions, and it's going to get worse. So I think we find the science matching the need, and we continue to work at it.
John,
Yeah, so again, I try to always turn to other disease areas for learnings, and cardiovascular space has now been well defined, right? And we know in that space that back in the 60s, there's an exploration of what was causing the increase in myocardial infarctions, and there was a huge debate as to whether it was cholesterol or thrombosis. In fact, I've got the papers where the pathologists were actually arguing this point. And when you went back and tracked the pathology of atheromas, you see the atheromas start presenting themselves 20 years before the onset of the symptoms, right? And we are currently looking at lipid agents, and we do know that there is a sweet spot for lipid agents, right? We know that once the drugs that target the monomer, let's call it, they don't work in congestive heart failure. It is too late then. They do work, however, still in the acute symptomatic phase. And so if there's any learnings or analogies here, I do think because the science is incredibly compelling, that there must be a sweet spot somewhere here for this disease where we're either targeting monomer or plaque. We should be able to have success. But again, it is trying to figure out from the trial design where is that optimum outcome measure and that optimum patient population.
Okay, Johannes and then Paulo.
So when I hear the question, on the one side is are we persisting, and then why A beta? So I think Samantha said it very nicely. We are persisting because AD has to be treated. And A beta, industry is not going only after amyloid. I would think everybody here on stage, we are not only looking at amyloid, we're looking in tau and all other mechanisms that are accessible. But all of you coming to this conference realize amyloid is even not solved from a scientific perspective. So when bapineuzumab, for instance, was started, it was a state-of-the-art trial. It targeted the population at the time. It even had amyloid PET in it. But we learned so much since then, and we are trying to do the state-of-the-art amyloid trials today, so targeting this very difficult area as state-of-the-art, going to the populations we think are right. Obviously, we are not all agreeing perfectly on the population, but everybody agrees that earlier is better. So I don't think persisting is blindly running in the same direction. It is trying to get the best molecules we're having into the best populations, and obviously looking at the best science to develop next generation molecules and mechanisms behind that.
Okay, Paulo?
Yeah, just a couple of additional thoughts. I think on the first hand, we keep tripping over ourselves because in its original concept, the amyloid hypothesis actually said that amyloid was necessary and sufficient. So therefore, if you treated amyloid, that's all you needed to do, you'd cure Alzheimer's. I don't think anyone here still feels that way. I certainly don't. Alzheimer's is an extraordinarily complex disease from a pathophysiological standpoint, and therefore, if amyloid works, and I do believe we're on the verge of seeing the first drugs that do work, it will work partially. It will not be a cure. It will be a treatment. It will have to be combined with other approaches, and therefore, but even that first step is going to be extraordinarily significant, not only because of what it will teach us about how to conduct trials for disease-modifying agents in Alzheimer's, but also because of the hope it will offer patients that actually you can slow down or alter the course of Alzheimer's. So I think we need to get away from that concept of a cure into more of a concept that targeting amyloid will be a significant step forward, certainly not the last thing we'll need to do. The second thing is that I think we've all learned in the past few years how complex even just A-beta is or amyloid is. And because A-beta is not one target. It's actually a sequence of targets. And up until very recently, and actually still now, we don't really know which one of these is actually the pathological species. And therefore, each of these medicines that you see target A-beta, they don't all do the same thing. And unfortunately, as I mentioned earlier, it just takes us too long to disprove each of the steps of that target. And I do believe now that we've eliminated a few of those, and we're focusing on some that appear to be more promising. Again, we won't make progress here unless we increase the efficiency of what we do, but I do believe we are now on the verge of something.
Great. David, did you want to make a comment?
I do think it's important to acknowledge whoever asked the question that there are a lot of trials going after amyloid, and it does feel sometimes like it's a kind of monolithic focus. But just to be explicit, and I think Johan said it nicely, in fact, what's really happening is there's a kind of an iteration in which there are different approaches and different trial designs and different populations that are being studied. And, you know, I don't think anybody knows at the end of the day, at this point, you know, whether or if any which will be successful. But it's not, but I don't think it's quite fair, I don't think it's fair at all to characterize sort of all the trials as chasing the same thing, because I just don't think that's what's going on.
Yeah, I think amyloid is certainly a central component in the disease. I don't think anybody should doubt that, as is tau and as is chronic inflammation. But I think we can perhaps now begin to question a bit whether it is sufficient to target amyloid as a sole treatment for the illness, because there is no one treatment for amyloid production, amyloid aggregation, or removal of amyloid. Just as there is, we'll probably find out there is no single approach to dealing with tau or certainly inflammation. So I think it speaks to us that we need to consider some of the other pathologies in the illness as well. And furthermore, this is an incredibly heterogeneous, incredibly complex disease. It's more of a syndrome, it's more of all Alzheimer's diseases that coalesce to a final common endpoint. And I think it remains to be seen where we intervene with a particular therapeutic modality at various stages of the disease. And I expect that as we become more adept at personalizing our approach to Alzheimer's disease, we'll find that certain approaches work for certain classes of patients, whereas others do not.
Okay, a corollary question then. If amyloid beta has a great deal of scientific justification, but it's not the only target, what are other non-A beta targets? And if the panelists want to comment about a particular non-A beta target that should be considered, please mention only one to let everyone who wishes to have a chance. Okay, you're up.
I would say that activation of the innate immune system in chronic inflammation is a very important target to consider. We've known that inflammation is part and parcel of the disease for a very long time. Fledgling attempts to treat it with anti-inflammatory drugs have failed. But nonetheless, as we learn more about the activation of the innate immune system in the brain, I think we'll be coming up with other targets. I mean, we look at RAGE, there are other targets as well. So that is, and it's a central factor in the disease, especially even related to amyloid pathology and also tau pathology. So that's one place where we're actively looking.
Anyone else?
So, John, I might be a very strong amyloid proponent with aducanumab and BACE in our portfolio, but I do think that tau is potentially the executioner of what amyloid is up to, and so we believe that those are very important pathways to understand. And there are multiple ways that we are looking at and should be looking at tau to try and work out what could be effective.
Great. Terry?
I guess the only thing I'll add, I know we're here to talk about disease-modifying therapies, but I don't think we should forget about symptomatic therapies. I think there'll be generations of patients that even if we have an effective disease modifier, it's unreasonable to expect that it's going to completely arrest the course of the disease. And so there will be needs for a better neurotransmitter-based therapies that can provide symptomatic benefit to patients. And I think often we forget about that. There's not as much research going on in that area, yet I think it's an area that's still of great unmet need.
Great, thanks. Andy?
Yeah, I was just going to add that I agree with that. And I think when we talk about symptomatic, we don't just mean treatments that might be able to improve cognition, but those that treat the many, many other symptoms that people with Alzheimer's disease have, including behavioral problems, sleep disorders, agitation, restlessness, irritability, psychosis, all things that we really understand very little about, but which require a lot of attention.
And take a terrible toll on the patient and the family. Johannes?
Yeah, so First, I don't really think we are the ones that should decide on the targets. I mean, the targets are really what is coming out of conferences like this. If I take what I take from this conference is the interaction. And if we talk about interaction, then we certainly have to talk about combination therapies. And I guess that will be, if we are now talking about an individual therapy, doing combination trials will be even more complicated. And if we look at the presentations here this week, we had synuclein and amyloid together, and tau and amyloid together, and amyloid plaque and synuclein and Lewy bodies. So there are a lot of different things that can will interact in different ways, and I guess that will be the targets of the future.
Great, thank you. David? No, okay. So another question, it's already been alluded to in many of the responses, since Alzheimer's disease is so complicated and complex and heterogeneous, should there be a precision medicine approach, patients stratified based upon sex, genotype, biomarker profile, other variables? John?
Yeah, so again, we're witnessing some cutting-edge science here in this meeting, and I can imagine me being in the hot seat 10 years from now, and someone saying, why did you not stratify based on a tau PET signal, right? Which seems somewhat intuitive currently. We've been, like I said, we want to make sure we learn from our failures, and so that each trial we do, we're advancing the science and the knowledge and the understanding. And so, one of the things that we currently have in both of our ongoing trials is tau PET, and it's not available for everybody and not available anywhere, but it will be a process of a learning step that when we get done, we'll be able to understand a little bit better. But right now, we don't really know it well enough. We don't know whether you want somebody with high tau for an amyloid therapy. Are they more likely to be driven by tau? These are really hard questions that we need the science to continue to advance, to learn. And so ideally, yes, we want to be able to stratify. We're doing that now when it comes to amyloid. We largely aren't enrolling amyloid negative people for amyloid trials, and I'm sure it will get further and further tailoring as the science advances.
Paulo?
Yeah, I think this is going to be a critical factor for success for all of us, and possibly one of the areas where it's going to be easier and most fruitful to collaborate between industry, across industry, between industry and academia, because certainly this is an area where we all need to work together. And I just reflect on the fact that, again, 5 years ago, we were not designing, the industry was not designing trials incorporating simple tests for amyloid. And that was a reflection of the fact that at that stage, we just did not have enough confidence in those biomarkers. It took things like ADNI and ABLE to convince us all that, yes, we can reliably identify people with amyloid pathology. And then it took a little bit while longer for us to be able to figure out how to operationalize those biomarkers into clinical trials that they could be inclusion criteria. And now it's become standard. It's a great first step. There's so much more. Tau imaging has been mentioned, but I think we're just really scratching, the surface. Alzheimer's disease, I'm very convinced, is not going to be a simple disease from a biomarker standpoint or from a testing standpoint until we really understand the complexity behind the genetics of Alzheimer and also the different phenotypes that within Alzheimer masquerade, and really add to the heterogeneity that we see in our clinical trials, we won't make progress. Now, there are initiatives trying to do that, and we've learned a lot, for example, from the familial Alzheimer's cases. But again, this is an area where I really feel that it's really not a race. It should be a collaboration.
Samantha.
Just to build on what Paulo said, confidence is one thing in prospectively, so at the start of the study, defining patients by a new tool. But then there are also some really practical issues. So if we take aducanumab in over 20 countries in more than 300 clinical sites around the world, implementing something new is quite difficult from the beginning of the trial. But one of the things that we do is continue to learn from conferences like this and add in some of these exploratory markers, such as tau PET, into the study as we go along. And so hopefully we can build in as much as possible in those types of clinical trials so that by the end, hopefully in addition to having a therapeutic, we would also be able to learn so much more about the optimal patient who benefits from these kinds of treatments. So while we stratify today, while we are all selecting our patients in more and more advanced ways, we do continue to learn in that cycle.
Okay, Johannes and then Larry.
Yeah, and I think in the question it was even going a little further, looking for other genetic marker, more highly selective. And we all would love personalized medicine, and I think we all can subscribe to that, get the right, perfect treatment for each individual subject. But if you just reconsider what Samantha said in terms of what we need for one drug, so we would be really happy to find something that does a little bit something on AD in general and then take it from there. And once we understand it better, most probably we can as well tailor it better. But we first have to understand it and then personally maybe personalize it.
Okay.
I think it's a great question. It's a difficult answer because what we do with biomarker work is in the course of a clinical trial, we in essence, prospectively validate that biomarker. We prospectively validate it in terms of expression of disease, in terms of symptoms. We prospectively evaluate it in terms of its predictive validity for clinical success. So we keep doing the experiment over and over again. When you start a phase 3 study, that program is put together, the protocol is written, the various procedures are put into the trial, and it's pretty well baked in. And so you're dealing with really scientific data that's current up to that point. And then you prospectively validate these things. So I think all the panelists here agree that this is a collaborative and iterative event. We won't get it right the first time, and we probably won't get it right the second time. But I do think that we're making progress in this. And so I think at the end of the day, when we see many of these trials, we're going to have prospectively validated markers that will allow us to pick patients who are likely to respond to a particular drug.
Okay, David, did you have a comment?
Yeah, sure. So to pick up a little bit on what Larry said, you know, and I think what everybody's been saying, there's a tremendous amount of learning going on. And there's a tremendous effort to really understand kind of what are the different currents within Alzheimer's, who are the different people. But, you know, I don't think we're at the point yet where we know enough to really be able to do you know, what I guess the question called personalized medicine. I mean, you really have to feel like you have a very strong hypothesis and a very clear understanding of who you're gonna, you know, take out of the general population and who you're gonna treat. I think maybe the closest to that, and actually, and amongst all of us, would be Diane, where in fact you can define a group of people who have Alzheimer's but differ from the broader population in a specified way, in whom you can begin to explore, you can explore a particular hypothesis. So I do think there is some of that work going on. I would expect that as we learn more, there will be more, but it has to be informed by what we actually know.
Okay, Paulo?
Yeah, I just had one final comment, which is to put a plug in for the importance of validating. The large majority of biomarker studies never get validated in this field, and by the way, in all of neuroscience, mostly because of small numbers. So if you're thinking about like genetic linkage studies, GWAS studies, everyone has been refuted time and time again, because again, the original studies are just not high quality enough. And that is an issue with the field until we start really making the effort of doing very large-scale genetic and genomic studies. None of these biomarkers will ever be as impactful as they need to be. The second plug is for the importance of the technology behind this. And to give you just one example of that, everyone's been talking about tau CSF testing for a decade. Until we started to develop a diagnostic test, the variability from lab to lab, from test to test, was as high as 20%. Getting it down to 2%, which is what our test now does, now enables those studies that get done to have real validity. So, you know, it feels like everyone, almost every other week, there's a new paper that gets published with an interesting gene or an interesting ligand, until we all come together as a field and really do the necessary validation work, not only on the tools, but actually on those large-scale studies, it will be very hard to make progress.
Great, thank you. So perhaps related question is, in the current trials or previous trials that failed, have there been subgroups of participants in which the agent seemed to have benefit? Does anyone care to comment about that? Subgroups of patients. Larry?
We see it all the time. There are people for whom the drug seems to work. I think the answer to why is sometimes unclear. It's dealing with an equation that has many unknowns. So, you know, is the drug actually penetrating the organ of interest? Is it engaging the target in that organ? Do you have appropriate biochemical engagement? Can you measure that engagement? And does that have any translation to the particular disease phenotype? And also, there are no two Alzheimer's patients who are alike. Certainly in my clinical practice over the years, I think in retrospect, they're all different. So it's really a matter of patient selection. It's a matter of how well your drug is engaging the particular target. And then I think the not only phenotypic, but also pathologic diversity, that no matter how hard we try in an Alzheimer's population, we still have a pathologically heterogeneous group of people. And it's, right now, I think we're getting better at parsing this out with amyloid and perhaps PET imaging, CSF tau A beta ratios, et cetera, et cetera, et cetera. But I think until we're able to parse those people out into pathologically distinct subgroups, we'll have a hard time answering that question.
Johannes? And then Andy?
Just a word of caution. We always have to be very careful when we look at a trial and it was not really what we had expected or wanted, and then we see a group in the analysis that does really what we wanted. Is there a good hypothesis for it? Is it pre-specified? Did we know it? It can lead you down a very wrong track if you just pick out those subjects that in that one trial did really well, but most probably it will not be replicated. So if there's a good hypothesis, go for it. But rather do it pre-specified, and if you have not pre-specified it, be very careful.
Okay. Andy?
Yeah, I think that's exactly right. What you're talking about is generating new hypotheses, and those need to be tested. And it's really another reason why we need to, as I tried to make the point before, why we need to do better phase 2 trials. This is the opportunity that we have to identify subgroups, to learn about whether there are subpopulations that might respond better or require different doses or require different concomitant therapies. And once you're doing this in phase 3, you have a serious problem if you end up with a subgroup that looks like it's done better, because now you're essentially back to phase 2 or doing another set of phase 3 trials. It's really a very difficult way to make progress in the field.
Okay, thank you. Several questions sort of reacted to the earlier discussion of the Diane Trials Unit, API, A4, and soon the early study, all in asymptomatic individuals. And the concern was, are companies still interested in treating people who are symptomatic? Are there going to be efforts to take care of people who already have the disease symptoms? Larry?
The answer to that is yes, a resounding yes. I think one thing that's instructive is, you know, if we look at other fields, a long time ago there was an autopsy study done in young men in Detroit. A urologist was interested of, do these young people have what looks like the pathology for prostate cancer? And some of these guys in their 20s and 30s, they did. These have been carried on prospectively. And so that it turns out that almost everybody over a certain age, every male here has got prostate cancer. It's in situ. It's asymptomatic, and it's going nowhere. But nonetheless, if you diagnosed it with a biopsy, then when do you treat? The same analogy, I think, can be taken to people with Alzheimer's disease. We know that people with autosomal dominant disease will eventually succumb to the illness. We know that people with ApoE4 have a higher age-adjusted risk for developing the disease. But treating an asymptomatic population based on what? And I think that that is, again, difficult for the general population. So we need to be able to say that can we treat people who present with particular signs and symptoms of the disease? And then secondly, I think the difficulty in treating the asymptomatic population is how in the world do you assess the effect of a clinical trial? Do you wait 30 years for them to develop disease? And what kind of outcome instrumentation can we use to look at success? And I think that that's unclear. Finally, despite our best efforts, even if we do develop drugs that slow the progress of disease or forestall its emergence, we will nonetheless have people who have symptomatic Alzheimer's disease. And we need to do something to help ameliorate those symptoms. I never had a patient who came to me who didn't want their symptoms fixed. I never had a caregiver that didn't want to see these behavioral symptoms ameliorated in some way. So I think, again, there's a bit of an inappropriate distinction between disease modification and symptomatic care, because I think it's a distinction without a difference in many ways. And so, yes, we are interested in treating people at all stages of disease.
David?
Yeah, I would strongly agree with Larry that the answer is yes. I might take a slightly different approach to it, though. I don't think it's dichotomous in terms of treating earlier and treating symptomatic. I think it goes to the question, perhaps there were a few questions ago when someone was asking about why are you guys all rushing at amyloid? And at least my answer would be that people are working on amyloid, working in different places, that amyloid, that hypothesis and that mechanism is being explored and tested through a range of places. We have the tools to test it definitively, and we're looking in different populations. At the same time, they were coming forward a whole series of other treatments. Some of the targets were mentioned here, particularly tau, some of the neuroinflammatory targets. Those targets are earlier. I think to some of the comments I made at the outset, it's important that we not take them forward until we really have the tools and the compounds that can actually test the mechanisms we want to test. But I think it's pretty fair to say that within the next few years you'll start seeing late-stage trials with some of the tau antibodies, hopefully with some non-antibody interventions in the same system. And there are a number of neuroinflammatory targets that are also coming forward. So I think there's a whole range of targets that are coming forward, and many of those, perhaps most of those, are I think, likely to be explored in patients who are symptomatic as a first entry into the clinic.
Okay, Johannes, and then Samantha.
So first to the question from the audience, and absolutely yes. I mean, Janssen is very passionate about treating symptomatic disease. Reminil has been developed, but a lot of you may as well know that D2 was a target in schizophrenia, and treating psychosis and schizophrenia was something Janssen was very passionate, is very passionate about. So yes, absolutely, symptoms have to be treated. I would like to react a little bit as well to my co-speakers here on the audience. So Larry asking on what basis would we treat? I mean, I do think that amyloid is the beginning of the disease. So somebody with amyloid in their brain biologically has the disease. If we think, for instance, about risk factors in cardiovascular disease, everybody would say somebody with hypertension has to be treated. So the risk if you have hypertension to get a stroke is significantly lower than the risk to get Alzheimer if you have amyloid in your brain. So I think having amyloid in your brain is a very good reason to treat. And I would like to applaud John and others to develop the tools. So I think Diane is a very thoughtful trial. The Diane group has been working on these very long. They really looked into the families, they developed the biomarkers, they looked into the cognitive tools. Now they're ready. That means you run the trials. Similarly, if we look at the A4 trial design, Paul and Risa Sperling have been spearheading that with others, using all the data from ADNI and ABLE, integrating it. I do think we can test these now. I think we have the trials now. Are these easy trials? No. No, everybody knows these are not easy trials. Early is a 4.5-year treatment trial. That is very difficult. But I think it's a time to test it.
Samantha.
Yeah, I would just like to add my voice to agree. The desire to treat patients with symptoms, absolutely, is important. We believe it's important. And we, all of us, I think, are united in saying that we want to do that. However, what keeps me awake at night is if we are in the scenario that the biology we are addressing requires that we treat that much earlier. And why that keeps me up is because maybe we've missed a generation of individuals who are symptomatic today. Our treatments aren't available for them today. But then maybe if we are in that scenario, the hope that comes out of it is we move the disease, that we don't see so many patients with symptoms. Now that's a bit of a dream scenario, as Larry said. People will still progress, we'll still see people with symptoms, but maybe less of them. And maybe the disease will look entirely different if we do have treatments that are started earlier, if they are more effective. But nonetheless, so it's a bit of a bittersweet scenario if we do find ourselves in it.
Okay, Paulo.
So my first thought is that actually we keep separating symptomatics from disease modifiers, whereas from the patient perspective, in terms of the symptoms or the benefit that these patients get, there's not such a difference. What we are doing with the so-called disease modification therapy is actually preventing the appearance of symptoms. And therefore, from a clinical standpoint, what you're actually doing is, at that end, pretty much the same. What we hope that we are demonstrating is that as the disease progresses, you are longer and longer into getting worse symptoms. So there's not such a difference. It's not like industry or pharma is not interested in treating symptoms. I would say conceptually we are very interested in treating symptoms generally. I'm even more interested in the situation where we can prevent the symptoms from coming in the first place. The second thing is that it's not that there's no interest in developing symptomatic therapies, but I think my realization in the past decade or so is that one of the reasons why we don't really have many new symptomatic medicines is that there are really not many new targets to look at, unfortunately. And several of the targets which are being developed have failed, either in phase 2 trials or in phase 3 trials, partly because there are standard of care medicines right now which are relatively good, and through better medical management, a lot of the symptoms are actually controlled easily. So what happens is that most of the new symptomatic therapies which are being tried have very modest effects on current standard of care. And that makes it really, really challenging because then you have to think about really very large trials to demonstrate a very small clinical benefit. So again, until we solve the biology behind this and understand really what are the new targets we need to be going at to, for example, to get better cognitive medicines or better symptomatic control of behavioral symptoms, right now, that's, I think, the main reason why there's not so much progress on that side.
Okay. David, did you have a question?
So I'm going to respectfully disagree a little bit with Paulo. I mean, so first of all, I guess I'm not so convinced that what we have today is really, it's great that we have something, but the something we have is maybe not so great. And I do think there are quite a few targets. I think, as Andy was suggesting, and I don't know if it was, maybe it was Terry earlier. I mean, I think as you think broadly across symptomatics, I think there actually is a large range of areas to go after. Sleep, psychosis, other behavioral issues, as well as cognition. There are a lot of cognitive targets. And there are a number of new things that are coming into the clinic at the moment. So I guess when I answered this question the first time around, I was thinking in terms of directly intervening in the core pathophysiology. But from a symptomatic perspective, for patients people who are already experiencing this disorder, there's actually quite a bit that's going into the clinic at the moment. And, and those trials are not impossibly large or impossibly difficult. So I'm a little more optimistic than Paulo is.
Okay, Larry?
I think we can draw some parallels with other branches of medicine. So, for example, we know that lowering hypertension has predictive validity in reducing cardiovascular and cerebrovascular disease. We do know that lowering cholesterol has predictive validity in prevention of stroke and heart attack. I think it is not entirely clear that lowering a beta or lowering tau or lowering anything in the pre-symptomatic Alzheimer's patient has predictive validity in emergence of disease. I hope that that will be the case, because I think that all of the work that's being done with, in all the studies that people have mentioned here, will point to a therapy that may have that effect. But right now, I would be a bit stumped to say, if someone had a positive A-beta scan and they're perfectly healthy, cognitively normal, and 45 years old, waiting for the emergence of disease in another 30 years, how, when, why, or should we treat?
John?
Yeah, again, I'll draw to the cardiovascular space. So we now know that the four major classes of medicines for coronary artery disease, right, to combine, they actually slow now the disease compared to in the 50s, 75%, which is a major impact. But what we do know is that the biggest growing area now is in the late stage cardiovascular end organ failure, congestive heart failure. So I would predict that, if we do have success in disease modifying, we have to keep our eye on the ball, because the symptomatic need will only grow greater than the current need is right now.
All right, among the currently available investigational drugs, are there certain combinations that seem compelling? And is it possible to consider combination therapy if none of those investigational drugs have received approval?
Yeah, so I think it's less a question of whether they've received approval, is just whether there's a strong scientific rationale for it. But, and so we can come up with good rationales, I think even at this point, for why we might want to combine different types of approaches. But it becomes difficult in the clinical trial setting to do this practically unless you've already shown some benefit from at least one of the agents. And I think that's kind of where we're stuck. I think once we have some success and there's something that we can build on, you're going to see a lot more in the way of combination treatments, because there'll be not only the rationale for testing them, but the ability to look and actually see whether you're achieving some greater benefit. So we look forward to that, but I think that's the reason that we're a little bit held up.
So some demonstration of efficacy.
Well, I would say at least for one of the agents. That would be, I think, a reasonable approach.
John?
Well, I mean, as a company who's published on some of the combined therapies, I think we're big believers in the need that we will eventually have multiple therapies that are needed in order to slow the disease. And, folks have argued, well, if we wait for them sequentially a little bit, to Andy's point, that it's going to be a long time. And that may be indeed the case. And that certainly is the most risk-averse way to do it. There is a pathway from the FDA's perspective to do combination therapies. It's called co-development, where you are testing two unproven agents. It is quite conceivable to do. And, we have today at hand some rational therapies, but none of them have proven, of course. And they even involve two amyloid types of therapies, like a monomer and a plaque. But obviously, we're now seeing the tau therapies enter. So clearly, an amyloid therapy and a tau therapy are also very rational combinations at this point in time. I do think that it is possible to run some of these trials, these phase 1, phase 2 trials. And while you probably won't have a hard clinical, say, cognitive score to make a decision on, one could make some decisions on biomarkers today on those impacts. But again, it will be a high risk area to undertake.
Okay, David, and then Samantha.
Yeah, so I have to say, I think there's some disagreement in the group here, because I'm much more...
Well, wait a minute. That means controversy.
That's good. That's what I...
Right. Okay, well, God forbid.
Go right ahead.
Thanks God. I mean, I agree with Andy, though. So you need two things to do combination therapy. The first is you need a good rationale. You need a very strong rationale. But the second is that you really need a development pathway. And having a development pathway when you haven't shown efficacy for either individual element is pretty challenging. The traditional way that's done once you get to the stage of having clinical efficacy, as opposed to showing PD effects being nice, but really just telling you can test the hypothesis. The way you do that is you test the combination together with each of the individual elements together with a control. And if you think about what a disease-modifying study is in terms of size and time and complexity, that's an extraordinarily challenging way to go. So it's not to say it can't be done, but it's very challenging. And I would argue, and you know, this is open to argument for sure, but I would argue the likelihood that a combination that a combination is efficacious when when you can't show with either of the individual elements some degree of efficacy it's on the more unlikely side of outcomes. It's not impossible for sure, and very few things are, but I do agree with Andy that I think a combination route is much more likely once you have something where you begin to show an effect and you then can begin to add on and to experiment with different mechanisms with it.
Samantha?
Yeah, so David has been very thoughtful about how to do the experiment properly. And if we go back to the conversation at the beginning of this panel, we were challenged about the experiments we're doing and whether we were persisting in the face of not learning enough. So the same thing applies to combinations. We do need to do those experiments very thoughtfully. And so just to lift up perhaps an example of what people might be thinking out there, two amyloid agents. One removes plaque, one's a beta-secretase inhibitor. These are often thought of as, yeah, sure, we should be doing combinations of those. Or should we do an anti-amyloid treatment and an anti-tau treatment? Well, it begs the question of, well, how would you assess whether the combination of the two of them would be better than any one of the agents? And I think you can break down each of those two stories. So there are no BACE inhibitors and anti-amyloid agents that have shown efficacy on exactly the same biomarker or exactly the same endpoint. So even the quick and easy experiment, which might be a 6 or 12-month experiment with a few hundred patients in PET, is still a very large undertaking, with one of them being a complete unknown. There might be circumstances that you do that. The clinical endpoint, as you've heard, for a monotherapy is upwards of 3,000 patients. So to do the design that David has just walked you through takes you up to 9,000, 12,000 patients. So the undertaking shouldn't be underestimated. So what we need to succeed is the ability to assess if those two mechanisms, when combined, do something that is additive at minimum, and something simpler to measure, something in a shorter clinical study. So obviously biomarkers is the one that we look to. Then if we take the amyloid and tau combination, which I think is tantalizing, how would we measure that the outcome would be better? Would it be on an amyloid endpoint, or would it be on a tau endpoint? So there are many unknowns, and entering into a clinical trial without a good hypothesis of what you expect to see in the endpoint really is not setting yourself up for success. So I think all of the panel acknowledge we'd like to do this. We think this is or will be the way forward, the way a complex disease is treated, but we need to make a few more steps of progress to enable that.
Okay, Johannes and then Paulo.
Very difficult to add to that, but just one small point. Lots of target, lots of great combinations. We don't even know what the sequence would be. So if you have these two great targets, do you do them both at the same time? Do you wait 3 months? Do you wait 6 months? Do you have to have a successful treatment? So as much as combination therapy is so obvious, I mean, you make a big point, it will be significantly more difficult, as David just said.
Okay, Paulo?
So I think I agree with most of what's been said. I mean, it has to start with a very solid mechanistic rationale. And, for example, thinking about the combination of two anti-amyloid agents, like a monoclonal antibody that removes plaque and a BACE inhibitor. We've actually published a few years ago with gantenerumab and a BACE inhibitor that you get significantly more, significantly faster. That's in an animal model, it's very encouraging, but what does that mean clinically? And that's where it starts becoming really complicated. Because then to actually conduct that experiment, you are talking about very significant large trials with unclear endpoints, and then you really have to wonder, what is it that you're trying to achieve? Is it the same clinical benefit but faster? Is it a higher clinical benefit? And then, and so what I'm trying to say is that there's a reason why, even though all of us, I guess, can conceptually agree with combination therapy as a desirable goal, we haven't really thought this through, and I don't think anyone here has the answer. To respectfully disagree with David a little bit as well, I actually do believe there is a situation where one of these agents alone does not work and only works in combination with another. And that is within the trial duration. If you're thinking about a trial that lasts 2 years, maybe one particular mechanism is not potent enough during that period of time to have a sufficient clinical benefit, but in combination, it adds to another clinical mechanism, and therefore the combination could be more powerful than the two agents alone. And that's an even more complicated trial design to do, because then you don't really know how to power that study. So again, I think conceptually we're all there, but to make significant progress, there is an additional need for further thinking.
We'll do David and then Larry.
Yeah, I was just going to say, I actually agree with you. So I started the controversy now.
What happened to that controversy?
Not enough controversy in this panel.
Okay, go ahead.
I think most of my colleagues up here are already doing combination therapy in a sense, because most of the patients in our clinical studies are taking a cholinesterase inhibitor plus or minus memantine. So in a sense, we're looking at a, quote, symptomatic drug in combination with drugs that we do believe will slow the progress of disease. The question I think that comes up with all of us is when we talk about adding two different mechanisms on top of standard of care, how do you assess, what do you call success? You can talk about biomarkers, but then again, the ultimate goal here is looking at some clinical success factor. And what would you call success? Would you call it additive? Would you call it synergistic? One just as good as the other. And I think that right now we don't know in any of our programs what success looks like with one agent. And so until we do that, I think it's very hard to start talking about combinations.
Okay, we're going to move on to another important question. Terry, I'm going to ask you to take a first crack at this, and it gets to that issue of sort of weighing efficacy versus safety. So we're all interested in dosing for the best efficacy, but that often brings the most side effects. So how is that determination accomplished?
Yes, I mean, I think that's sort of fundamental to really any drug development program, is sort of the whole risk-benefit question. And I think, specifically thinking about anti-amyloid therapies, one of the big questions is how much A-beta lowering do we need? Because it's still a very, very much an unanswered question. So, is 75% reduction from baseline sufficient in A-beta 42? Is 50 good enough? Is it different depending on whether you're in a more advanced patient population versus in a more of a secondary prevention paradigm? I think that's sort of a fundamental question that is really currently unanswerable. And so you're left with going in with really trying to drive to a maximum dose that you feel can be justified by safety based on the data that you have. sort of a no regrets dose, but there's danger in that because of the duration of these trials. You could end up with a safety finding that you really wasn't predicted by earlier therapy. So you really need to be able to have a lower dose as part of these efficacy trials to sort of safeguard against that. And I think that's really one of the major drivers of understanding how you balance it. It's really a difficult question because you don't really know what your target for efficacy is at this point.
Thank you. Andy?
Yeah, I just wanted to say that it's as Terry says, it's a very standard question in drug development, obviously, that we look at the risk-benefit ratio. But in this case, it's interesting that it opens up a window into some very interesting questions that have come up, I think, during Alzheimer's drug development particularly, that are worth thinking about. For example, I think it has been suggested that we may consider whether the relative safety of the drug might be different, or that we might tolerate a different relative safety of the drug for a preventative therapy than for a symptomatic therapy, or for a therapy that we're using in, quote, younger people or people who do not yet have symptoms versus people who already have symptoms of Alzheimer's, who are Alzheimer patients. And I think that's a very complicated thing, and I'm not sure that we have really paid enough attention to some of that, and I'm not prepared to comment too much further on it right now, but I just think in that sense, it's a very interesting question.
Okay, Johannes, and then David.
Yeah, Andy, you go into a very interesting direction. I mean, obviously, from a development perspective, and specifically in the area range where we are currently now, we have no clue what the effect size of our treatments will be. So how much clinical benefit will we bring? And in that sense, it is very difficult to assess how much risk we think is ethically correct, because that is finally the question, is it? So how much benefit can you bring with the therapy, and how much risk are you willing to have then together with it? And I see Maria here in the audience, and I think it's coming to the patients, is it? So we have done preference studies asking healthy elderly that then with the risk, how much risk would you be willing to take to have Alzheimer 2 years later, 3 years later? And it's incredible. People don't want Alzheimer. So if you ask patient subjects what they are willing to take as a risk, it's certainly much more than any of us here would ever be willing to move forward in a clinical trial. And I think it's ethically extremely challenging in a field where we yet do not know what the treatment effect will be to bring that into the development perspective.
Okay, David?
Yeah, so I think this is a hard question, and I think that in particular the question comes up now and is maybe more urgent in the context of studies of people earlier in the disorder before they have...
David, hold it closer.
Before they actually get to having symptoms. And so just a little bit of history here. So when we started the 8931, the Vera Basisat studies, we actually made a conscious decision to run the symptomatic studies first, that is the mild to moderate study first for a year prior to starting the prodromal study. And the reason was really that, the prior that we had, the Bayesian prior, if you will, was really semaglutide, where people got worse, right? And I think the assumption and the belief has been that those are off-target effects. I'm not sure anybody really knows that or can prove that. And so what it does or ought to do is remind us that any clinical trial that starts at equipoise. It starts at therapeutic equipoise, meaning, and which is why you do two-sided tests. You have a treatment that may make you better, but you also have a treatment that may make you worse. And that, I think, gets very complicated as you go earlier, where you think about, for example, a treatment that, say you ended up with a treatment that made people worse by, say, 2 points on the ADAS-Cog. Well, now you've taken essentially 6 months of health, or you've accelerated towards the disease by essentially 6 months. So there really is an obligation, and I don't know what the right answer is, and I think Johannes has talked about it, and Andy's talked about it, but it is a very complicated problem, and we have to remember, and I think the first thing you have to remember is that you do start there's something called the therapeutic misconception, which is that, oh, you have a new experimental treatment, it must do something, it must make people better. But that's not, in fact, where we start. Where we start is with a new treatment that could be better, could do nothing, but also could be worse.
Thank you. John?
Yeah, so I mean, I think a priori, right? You must prioritize safety over the benefit, because the safety is often known, or you're seeing that signal, and the efficacy is unknown. So, the question you posed is a loaded one and somewhat of a false security, because the question is how much risk would you take to delay your AD by 2 years, and that implies that there is a delay of 2 years, right? If you ask the question, how much risk are you willing to take, and you don't know whether there's a benefit, you may get a very different answer. And so we don't know the benefit side of things. And so I do think we have to be a little bit cautious, because we haven't had a lot of success. So the posterior probability of success on the efficacy side is really, really quite poor for us so far. And so trading that off has to be not done lightly, obviously.
Okay, Johannes.
Just reiterating, I tried to say that as well. I mean, as long as we do not know the effect sizes or the effect at all, we have to always be careful. But imagine, and I think where the question is we're coming from, if you would have a successful treatment, even then we may think about it very differently than patients would think about it.
Okay, so I have a number of questions from the audience that had a lot of support, very popular. And we'll see who wants to address this first one. We've heard from our panelists that our current phase 3 trials have been designed several years ago and don't keep up with the most advanced science of today. But based upon what you've learned from this ADPD 2017 meeting, what would you do differently now from our current phase 3 studies? Has there been something that has, or several things that you now would like to incorporate in design of future phase 3 trials? And don't answer all at once. So. All right, Larry's going to pick up.
I'll take a futile stab at this thing. Our phase 3 trials are just about enrolled. They're in the book, and we're not going to go back and change them. But, if I could turn back the clock and say, what would we have done different? I think we could have used more refined diagnostic techniques. I think that we also perhaps could have done something apart from a somewhat slavish reliance on some of our clinical outcomes. Alzheimer's disease is not necessarily a disorder of the ADAS-Cog and CDR.
Well, wait a minute, this is controversial.
I did that just because they're good tools. And I also think that we probably should be looking more closely at a large number of biomarkers, not just simply the usual suspects involving A-beta and tau, but also looking at inflammation and validating these prospectively. Again, a prospective validation in the course of a clinical study is not going to help you get registered apart from maybe making some noise about target engagement. But it would be good for the field in general.
Okay, John?
Yeah, I mean, I think I alluded to this a little earlier, that 10 years from now I'll be sitting here defending decisions that we made today. But I mean, clearly, I think the tau data is very compelling. It reminds me a bit of the stroke field. It's very asymmetric. Where the tau is looks like where your deficits also tend to be highest. And certainly today, I wish I knew the answer about how best to utilize that biomarker, because I do think we could do better enrichment and identify the patient better. I don't know yet. Today, I can tell you how to do it. I wish I could. I think the other interesting thing that I find intriguing today is the rise of neurofilament light chain, which is an important biomarker that seems to be rising in prominence. We've seen it have some predictive validity in MS trials, even for even therapy. And then I think I want to point out another thing that Paulo mentioned in passing, and that is Roche's CSF assay. I think is a huge step for our field in getting some kind of ubiquitously translatable assay that we can use in these worldwide trials. I mean, today in Lilly's trials, we do allow CSF, and they all get shipped to one single site, and they get batch processed. It's a long process to enroll those patients, just because we don't have confidence in laboratories around the world. And I think this Roche assay will do a lot to facilitate that. And so it is one of those things I think can be implemented today that's a major improvement for clinical trial performances.
Okay, Paulo?
Yeah, so maybe the reason why I guess the audience sensed a little bit of hesitancy in the, a little bit of hesitation in the original response from the panel is that the way you framed the question, John, was about what would you change in your phase 3 trials. That's not something we want to hear, because phase 3 trials are pretty much locked down the moment you start them. And if you start...
I misspoke. I think the questioner wanted to know, going forward in your future design, what did you learn from this meeting that you then would incorporate?
Right. So to that point, what I think I learned at this meeting, as in several other meetings, is that there's always very exciting new science bubbling up, either from preclinical models or biomarker studies. You know, neurofilament came up, tau came up. We need to start doing now the validation work to make sure that those are going to be ready for the next generation of trials. And that's not an easy process, as I tried to explain. It takes years of doing that. It takes replication and validation. And unfortunately, it's not very sexy work to do, but it's crucial for quality. And that is what you then need to have to incorporate these things in phase 3 studies. So what I've learned here at ADPD are going to be some of them things that then we need to pursue and make sure that we explore, that we validate to be ready for the future.
Great. Samantha and then Johannes?
So I'm sorry if I left the impression that we never change a phase 3 trial. Obviously, echoing Paula, the cornerstones of the study we're not going to change. But there's a lot of opportunity for us to collect samples, in particular fluid samples that can be applied to biomarkers that are discovered and validated during the course of these long studies. And so we are definitely continuing to look at the discoveries in those space so that at the end of the trial, these studies can be the new breaking science in 2 years, the next ADPD and the ADPD after that. So we do have the opportunity to implement some of these new scientific findings, and so we are looking at many of those. The one thing that I would say, what would I have done more of, is also around biomarkers and tools. I think the tau imaging space is more confusing than earlier, at least in the bit that I dipped my toe into here, and the sub-study that we're doing, perhaps I would have done more ligands if they were available, because this idea that putting an exploratory biomarker into a phase 3 is concurrently validating the tool as well as the finding may mean that we need to do more of them in the context of those clinical studies. So that's what I would have done differently.
Okay, Johannes?
So most certainly I'm a little bit blindfolded, but I feel for early on the A4 platform. This is what we can do currently in terms of population. And we have potentially changed too much on the way moving into, but I don't think we can change much. I have a wish list though, and the wish list is very much aligned with what the other speakers said. So I would wish there would be a universally available tau tracer where everybody knows it measures and everybody agrees that's the one tau tracer we want to use. I would wish there would be a CSF-A beta assay that is universally available and we could use in every site, and the same for an amyloid PET. So obviously, if the tools would have the accessibility and availability and validity, that would be of huge help and would help us to do these trials. Coming out of this meeting, I think the wish list grown significantly longer. Unfortunately, we cannot act on all of these right now.
Okay, Larry and then Terry.
I think the wish list continues to grow, but I think all of us, to one degree or another, are participating in the process. When we collect biomarkers in the course of a clinical study, we are prospectively validating them with not only in regard to the mechanism of the drug that we are looking at, but also with the clinical progress of those patients who are treated, and perhaps for the field, more importantly, with the progress of placebo patients. We share those data with others. We work with ADCS. We work with ADNI. And so what would we add? Well, right now we're trying to figure out what we should add for the next generation of phase 3 studies. And one of the important things, I think, for all of us here, and industry is probably one of the very few places where you can do this sort of validation work. It's the only place where we've got the trials that are big enough, that are long enough, that involve enough patients across a number of different treatment modalities to collect both fluid phase biomarkers, to look at imaging biomarkers, assess their predictive validity, and then turn that crank, as it were, back into how they can have some utility in the next generation of studies.
Okay, thank you, Terry.
Yeah, I think at this meeting and others, it's increasingly more apparent, I think, to myself that we do need to go earlier, at least with a BACE inhibitor or a GSM, which is, again, what we have in play at Pfizer. Something that didn't come up at this meeting, but certainly would be interested in hearing from the other panelists on, but something that's going to sort of keep me and others at Pfizer up at night is as we go into some of these secondary prevention type of studies, it's a very different, very big paradigm shift in terms of recruiting and thinking about the massive amount of screening that has to happen. And obviously there are different initiatives throughout the globe to try to support that. But I think it's a tremendous undertaking, and I think that's the one thing to try to enable these, and there'll be many, I think, of our companies that might be exploring going into this earlier population, and how we're going to be able to accommodate and recruit these studies, I think is a big thing, and I certainly would love to hear from Johan on that and others.
Great. So there are many questions that have been submitted. They're all terrific and they all are very germane, but we just can't address them all. So I'm going to come back, I think, Samantha, in the opening remark portion of this. I think you were discussing as we move the field forward that there will be opportunities to be beneficial for companies to collaborate, and in the spirit of collaboration, I'm going to ask each of you, we only have a few minutes, so succinctly, if there is an investigational drug that is sponsored by a company other than yours, which one do you think might have the highest chance of success? So, Larry, you're up first. Succinct.
These people are my colleagues and friends, and I wish them all well. And frankly, I don't know. I had great hopes for beta secretase. I think that will still prevail perhaps in earlier disease. And the antibodies, I think, for me, are still an open question.
John, I'm a little bit conflicted, having touched many of these molecules. So as a mechanistic class, beyond the molecules in my own portfolio, I do have a soft spot for the science of beta secretase in addition to antibodies.
I guess we're all kind of conflicted here. So I think that the most likely mechanisms to work in the next few years are going to be the antibodies against amyloid. And beyond that, I think that I'm really curious to see what an anti-tau drug does.
Yeah, I mean, I think, it's a hard question. I don't know what I think. No, I mean, I think if amyloid if anti-amyloid therapies are to work, they're most likely to work earlier. I mean, I think that just makes sense based on the evidence. We're not doing that study at the moment, so I can say that, right? And beyond that, I think in people who are already symptomatic, I actually think the tau and neuroinflammatory approaches, which are only just beginning to come forward, are actually probably the most interesting.
Well, I'm the only one who can easily hit a home run here. Obviously, I think that our esteemed partner Biogen's drug is going to be successful. And beyond that, we're quite excited about the upcoming tau approaches as well, which I think are have a good chance as well.
So just specifically, it's aducanumab, the
mentioning-
anti-amyloid antibody.
That's
if you had to
correct,
pick.
aducanumab.
Okay.
Yeah, so I'm biased. Obviously, I work on BACE inhibitors. I do have a strong hope that actually all the BACE inhibitors, which are very similar for the most part, I think what I wish for is what I think what the world needs, and that is I think a pill is cheaper for the world. It's easier to do, and it's also easier to take into these prevention studies. So my hope is that the BACE inhibitor class is a successful class.
So I really do hope that one of these trials will be successful and we will get something that will help patients. I'm currently very much trying to motivate my mother to do something because I think she should. So in terms of the class, I think I don't care too much what had the biggest chance. I think if we look at the current data, really science, aducanumab seems to be the front runner, but GAN seems to be very close to that. So my thinking is that these two may be very close.
I guess I just have to go with what evidence there is, and the best evidence that I've seen out there so far of the things that are in phase 3 is aducanumab. So that's what I would probably put my money on if I was betting.
Great. So thanks to everyone who submitted questions, but will you all join me in thanking our panelists who all were terrific. Thank you so much. And with that, we will conclude. So thank you all for attending and enjoy the rest of the meeting.